Good morning, students. Today we are going to continue our discussion about the topic recombinant DNA technology. In the last class, we discussed about the various steps in gene cloning or recombinant DNA technology. We also discussed the definition of gene cloning. Let us proceed forward today with more details about this technology. Today, what we are going to discuss is tools in recombinant DNA technology. Tools in recombinant DNA technology. So, to do any kind of gene cloning, we need to have certain tools. And the tools can be broadly classified into two categories. One is vectors and the other we can classify them into enzymes. So enzymes and vectors are the most important tools in recombinant DNA technology. Today our focus is going to be on enzymes. Let us start with one of the most important group of enzymes, which are called as nucleases. We are going to start at the topic of enzymes with nucleases. So what are nucleases? Nucleases are enzymes that can degrade, cut or shorten nucleic acids. So nucleases are enzymes that can cut, degrade or shorten nucleic acids. What do we mean by nucleic acids? We have two types of nucleic acids, DNA and RNA. So enzymes which can degrade, cut or shorten these nucleic acids are called as, are broadly classified as nucleases. Okay. Now, even among nucleases, we can once again, depending on the specificity of the nucleases, we can classify them into DNases and RNases. The names are very clearly telling you what they are. DNases, they break down DNA. RNases break down RNA. Or they cut, DNases cut DNA, RNases cut RNA. So depending on which molecule we want to cut, we can either select DNase or RNase. Now, DNases, once again, depending on their action. Depending on the action of DNases, we can again classify them into exonucleases and endonucleases. Exonucleases and endonucleases. So let us concentrate on this term exo. Exo literally means outside and endo means inside. So exonucleases are DNases. Remember, they are under the classification of DNases. Exonucleases, what they do is whenever they find a DNA molecule, so imagine this is a DNA molecule. This is a chain of DNA molecules with a lot of nucleotides. Each one of these circles represent a particular nucleotide. Okay? So what exonucleases actually do is that they simply attack the DNA molecule from the outside. They can only attack the DNA molecule from the outside. Okay, so what do? They attack the DNA molecule and they cut, I'm sorry about that, they cut the phosphodiester bond. Remember, each of these nucleotides are held together by a bond called as phosphodiester bond. So DNases simply degrade this phosphodiester bond. Alright, so once the phosphodiester bond is broken, the DNA molecule reduces, shortens in length. If one nucleotide is removed, it is shortened by one nucleotide, or if a couple of more nucleotides are removed, it gets shortened by so many more nucleotides. So once again, exonucleases can either attack single-stranded DNA, or the DNA can even be double-stranded DNA. You have both types of DNA, and exonucleases can attack both these types of nucleotides. Okay, so here we are seeing that the nuclease is going to attack the double-stranded DNA, and this is how it's going to cut, and the DNA gets shortened by a pair of nucleotides. So I hope you have understood this. Nucleases start removing the nucleotides from the ends of the DNA molecule by breaking the phosphodiester bond. Okay, whereas endonucleases, they are the opposite of exonucleases. So what they can do is that they can simply break the DNA molecule anywhere in between. Supposing here. So what has happened? You've got two strands of DNA. Now this was attached here. The endonuclease attacked this phosphodiester bond. This got broken, and you have two pieces of DNA. It can furthermore attack these fragments of DNA. Cut this here. Alright. So in the next step, cut it here. So you get individual nucleotides like this, and we say the DNA has got completely degraded. So either they can cut the DNA, they can shorten the DNA, or they can completely degrade the DNA. So the same with exonuclease and the endonuclease. They can do any number of cuts. It's not necessary that they just go and cut once. So once this is removed, it can break this, remove this also. It can break this, remove this nucleotide also. But exonucleases do it from one end or from both the ends, whereas endonucleases can cut the DNA anywhere in between the DNA. So this is the basic difference between exonucleases and endonucleases, which are two types of DNases depending on the action or depending on their action. Okay, so let us take in more details about these DNases. We as biotechnologists use a number of such nucleases. In a number of cases, number of instances, number of experiments, they are used to degrade and to cut DNA and RNA molecules. Alright. Anyways, now moving on to a specific type of endonuclease, which we biotechnologists are very interested. This is called as restriction endonucleases. So in this class, let us try to understand the basics about restriction endonucleases. Now let us look at the name. This part, I'm sure you are clear now. It is first of all a nuclease. So it can break down nucleic acids. It's an endonuclease. So it is an enzyme which is going to cleave or cut the DNA from anywhere inside the DNA molecule. Now let us try to understand what is the meaning of this restriction. Why these enzymes are called as restriction endonucleases. So let us take the definition first. They are enzymes that can cut double-stranded DNA at a specific sequence that it recognizes. So they are enzymes, restriction enzyme, endonucleases are enzymes that can cut double-stranded DNA at a specific sequence that it recognizes. Whereas normal endonucleases, what do? If this is a DNA molecule, if you add an endonuclease, I am drawing a double-stranded DNA. If you add a general endonuclease, the endonuclease may cut the DNA at this point, or may be yielding 2 fragments like this. Alright, and next time it can cut it here, so you get 3 fragments like this. Alright, next time it can cut it here, so you get 4 fragments, and so on. But if to the same DNA molecule, if you are going to add a restriction endonuclease, alright, I'll call it RE, restriction endonuclease, if there is a specific sequence it recognizes, only if there is a specific sequence that the restriction enzyme recognizes, then it will cut the DNA at that point, like this, and it won't cut it any further. Supposing this sequence is not there, then the restriction enzyme will just leave the DNA as it is. It will not even cut that DNA. So that is the definition of restriction endonucleases. Let us look at it again. They are enzymes that can cut double-stranded DNA at a specific sequence that it recognizes. So specific sequence that it recognizes. Is that clear? Moving on, where do we find such restriction endonucleases, or how did scientists come across these restriction endonucleases? Restriction endonucleases, they are found in bacteria. Most bacteria, I'm sorry about that, most bacteria produce these restriction endonucleases. Alright, and these enzymes have the ability to cut the DNA molecule at a specific sequence. Alright, so let us try to understand its function inside the bacterial cell. Or rather, why the bacteria is producing these restriction endonucleases. There are 2 functions of the restriction endonucleases inside the bacteria. Number 1 is that whenever, imagine this is a bacteria, this is its chromosomal DNA, and this bacteria is producing some restriction endonucleases. Alright, supposing there is a viral attack, a bacteriophage attacks this bacteria. It is inserting its DNA inside the bacteria. Alright, now these restriction endonucleases have the ability to go and degrade this viral DNA. Why? Because they can recognize specific sequences on the virus. So as soon as the viral DNA has entered, they go and degrade the viral DNA. Okay, they degrade the viral DNA when it enters the bacterial cell. Once the viral DNA is degraded, the infection is no longer able to proceed. Otherwise, what will happen? The viral DNA takes over the entire cell and degrades the chromosomal DNA of the bacteria and uses this bacteria to make more of these viral particles. Finally, it will break down the cell, rather kill the cell, and more number of viruses are released out. Okay, but now what has happened? This is like a defense mechanism of the bacterial cell. It is producing restriction enzymes which go and degrade this viral DNA. Thereby, what is it doing? It is restricting the entry or the propagation. Propagation means division. Propagation of virus inside the cell. Inside the bacterial cell. So this is a fantastic function of the restriction enzyme. The bacterial cell is able to protect itself from the virus, and what is happening is that it is able to restrict the propagation of the virus inside the bacterial cell. Now, second function is, automatically we can ask a question. What has happened? This viral DNA has entered the cell. Restriction enzymes go and attack and break down this DNA. What about the bacteria's own DNA? This is also the same as the viral DNA. Viral DNA also has the 4 nucleotides A, G, T and C. Bacterial DNA also has the same 4 nucleotides A, G, T and C. So, if these restriction endonucleases can degrade this DNA, why can't it attack its own DNA and degrade that also? Isn't that a question, logical question that comes to your mind? Definitely yes, because both of them, ultimately the chemical nature is the same. Both of them are DNA molecules. So how is the bacteria's own DNA escaping from the action of these restriction endonucleases? Again, that is due to the modification function of the restriction endonuclease. The same restriction endonuclease which is able to restrict the propagation of the virus inside the bacterial cell also modifies the bacteria's own DNA, modifies the bacterial DNA. How does it modify its own DNA? So wherever it has its specific sequence, it acts, it methylates its specific sequence at one of the nucleotides. Okay, let me explain this to you. What this enzyme does is, supposing the same sequence that it is recognizing inside the virus is present in the bacterial DNA. Okay, what it will do is, it will add a methyl group. Okay, it will add a methyl group to one of the nucleotides in that specific sequence. Now, supposing this is a specific sequence. For example, let us say this is a specific sequence that one of the restriction endonucleases is able to recognize. Okay, GAA-TTC. Alright, so what will it do? In its own DNA, wherever GAA-TTC is present, it will go and add a methyl group to one of the nucleotides. In this case, it has added the methyl group to one of the adenines in the specific sequence. So now what will happen when its own restriction endonuclease comes, it will not be able to recognize this sequence because there is an extra methyl group sitting on the A there. Okay, so that is why it is able to protect its own DNA, protects its own DNA. Whereas viral DNA does not have this methyl group, and therefore restriction endonuclease can go and cut this DNA, viral DNA. So you can see there are two functions of the restriction endonuclease inside the bacteria. One is it is restricting the propagation of the virus inside the bacterial cell by degrading the viral DNA. This is also called as the restriction function. This is called as the restriction function, and this is called as modification function, wherein its own DNA, its own bacterial DNA is getting modified in order to protect it from degradation. So these are the two functions inside the bacteria. Therefore, this system is also called as restriction modification system. Restriction modification system, or RM system, because the same enzyme is able to do both restriction function and modification function. Okay? Alright, so restriction enzymes were actually discovered by a set of scientists. They are very important scientists. They also received the Nobel Prize for this discovery. W. Arbour, H. Smith, and T. Nathans. So these are the scientists who discovered the restriction endonucleases. Arbour, Smith, and Nathans. They discovered the restriction endonucleases first and in a bacteria, and after which more than 250 restriction endonucleases have been discovered in many different types of bacteria. There are different types of restriction endonucleases. They are simply called type 1, type 2, and type 3. Among the three, type 2 restriction endonucleases are the most important restriction endonucleases used for genetic engineering or recombinant DNA technology purpose. Alright, the reason being they are also called as molecular scissors.